Hi, my name is Joel Nitta. Thanks for coming to my poster, an open and continuously updated fern tree of life. So this is the poster that accompanies my talk, and you can find the link for my talk in the upper right-hand corner of the poster. So if you get a chance, please do check out the slides for the talk as well. A major goal of biology is to build a complete tree of life. One way to do this is to use data on GenBank, but there are some challenges with this. First of all, trees built with data from GenBank will go quickly out of date because of the rapid accumulation of data on GenBank. And secondly, automated pipelines may often incorporate misidentified species or other problems in the tree. So the goal of this project is to construct a mostly automated pipeline to build a taxonomically high quality, maximally sampled fern tree of life. Now you'll notice I'm not trying to build the entire tree of life. There are some other projects that are seeking to do that. Rather here, I'm focusing on just my study system, ferns. So this is an overview of the workflow that we use to build the fern tree of life. And I'm just going to go ahead and walk through some of the main features that I think are really useful and might be helpful to others. First of all, we increase the efficiency and reproducibility of working with GenBank data by using a local database. And this is because we need to query the GenBank data multiple times, so it's more efficient to use a local database. And furthermore, it can increase reproducibility because we have a snapshot of GenBank data, and otherwise data on GenBank are being updated every day. Second, we extract sequences from the GenBank data without relying on annotations. And that's because there's no standard format for gene annotations in GenBank, so the same gene could be annotated in several different ways. Instead of that, we use a BLAST search against a reference database and then extract homologous regions. Furthermore, we implement a custom fern taxonomy, and that's because the NCBI taxonomic system was not designed for ferns and it includes a lot of synonyms. So by resolving the names in GenBank to our custom fern taxonomy, we can output a tree that is really high quality and can be immediately used by researchers working on this group. Furthermore, we use a two-step phylogenetic analysis to maximize both accuracy and sampling. And you can see that in the workflow on the right. So the top part of the workflow starts from whole plastomes, these blue circles. And the bottom part of the workflow starts from Sanger sequences, which are shown by these orange lines. Now, in both cases, we extract sequences, resolve taxonomy, and then concatenate these into alignments. And we first then use the plastome sequences to build a backbone tree. And this backbone tree is used as a constraint to do phylogenetic analysis of the Sanger sequences and generate the full tree. Now, by taking this two-pronged approach, we can both generate a tree that is robustly supported across the backbone and then maximally sampled at the tips. And finally, we incorporate both automated and manual checks for rogues or misidentified taxa in the tree. And in the workflow, I've indicated steps that are either manual or partly manual with the hand here. And for example, near the end of the pipeline, we actually manually inspect the tree. And so prior to doing the final phylogenetic analysis, I'll do a phylogenetic analysis in fast mode in IQ tree and inspect that tree and look for any mis-IDs. So that's an example of a manual check. A more automated check is doing an all blast search. So during that check, we do an all by all blast search. And if the query sequence matches the wrong family, then we'll exclude it as a possible contaminant. And I do inspect the results of that before accepting them. So that's why I've also indicated that with the hand here. So the results of this workflow, we were able to sample about 5,500 fern species out of an estimated 12,300 extant species. so almost half. Now, this is a 40% increase relative to the previous largest fern phylogeny. Interestingly, our divergence timing analysis suggests that compared to previous studies, ferns may have diversified earlier than previously thought. Now, importantly, we don't want to publish this tree just once and consider it done. We've designed this pipeline to be continuously updated, and we are always pushing our results up to a web portal to make them available to other researchers. So that's here at fernfly.github.io. And of course, all of the data and code underlying our tree are also available on GitHub and Figshare. And we also have an R package that can be used to read the tree directly into R as well. So future directions for this project. I want to complete the final fern tree of life, finishing that unsampled 50% by sequencing herbarium specimens. And I also want to integrate the fern tree of life with the upcoming Pteridophyte Phylogeny Group 2 taxonomic system, so that we can have a taxonomic system which can be a living online document that can be updated as new phylogenetic hypotheses become available. So with that, I would like to thank all of the organizations and individuals that made this research possible, and you for listening. Thank you.